and severe seizure, epileptic seizure. So first we studied why the MCD occurs. And we analyzed KIF2A conventional knockout mice. And you can see this is a knockout and wild type. And you can see in knockout neonatal onset seizure with clown. You see here? Seizure occurs. Okay? And so even after 6 hours, knockout mice failed to roll over with the coordination disturbance. Okay? So we found out the small pale body and congenital microcephaly in conventional knockout of KIF2A and the cerebellar hypoplasia. As you can see, wild type and knockout, in knockout mice, you can see lateral ventricular dilation and a thinner cortex and the superior colliculus, and hypoplasia of the cerebellum and midbrain. So microcephaly occurs. And this is the cortex of wild type and mutant, and cortex of wild type and mutant. You can see clearly laminar defect. Also you can see in the hippocampus, in knockout, laminar defects occurs. And obviously corpus callosum in knockout is much smaller. So we studied the reason. The reason is the apoptosis occurs in a knockout mice brain here. So this is called the loss of neuron. So this is a summary of the MCD-like phenotype of KIF2A knockout mice. Neonatal onset seizure and thinner cortex and superior colliculus and hyperplasia in cortex, smaller corpus callosum, and so on. And the apoptosis occurs. So next, why epilepsy occurs? The mechanism of epilepsy. To study postnatal function of KIF2A, actually KIF2A is abundantly expressed in postnatal. We generated conditional knockout. We, using tamoxifen, we conditionally knocked out KIF2A postnatal. So we applied tamoxifen from 2 weeks postnatally, about for a week, and completely deleted KIF2A postnatally. Then what happens? So conditional knockout mice show severe epilepsy, as shown here. Okay. You can see severe seizure. Okay. So where is the locus? So we found both cortex and the hippocampus, the severe seizure occurs. I show you one video. It's a cortex-oriented epilepsy. So during this seizure, we recorded electroencephalogram by this process. Okay, and we found also in hippocampus the seizure is originating. You can see here. Obviously, these knockout mice showed a severe seizure, epilepsy. Okay, so what is the mechanism? This is the wild type and the conditional knockout stained with KIF2A, and also this is the wild type and the conditional knockout stained with neurofilament M antibody, which is a marker for axon. As you can see, comparing with the wild type and conditional knockout, there are aberrant axonal network here in this region, very abnormal. So we thought aberrant mossy fiber sprouting occurs and form recurrent network here, as shown here. That is true. If you culture granule cell, this is wild type, and conditional knockout, many long branch of the axon emerge. This could form recurrent circuit. And if you express exogenously KIF2A, then you can rescue this phenotype. So this happens clearly because of the loss of KIF2A. Then we performed electrophysiology and proved that the HeLa stimulation caused paroxysmal depolarization shift in conditional hippocampal slice culture. So obviously recurrent circuit, abnormal circuit is formed here. That was confirmed by the staining with zinc. Zinc is enriched in mossy fiber axons. So compared with wild type, knockout, there is lots of axonal network here, abnormal network. So that another data by the YFP expressing mice, conditional knockout mice, show clearly in this layer there are aberrant abnormal network of axon. So as shown here, this is wild type, this is a conditional knockout stained with neuronal marker, neurofilament L. You can clearly see in this region compared to the wild type, there are abnormal network of axon. That was proved by the single neuron level. This is wild type, and wild type neuron extend long axon, one axon, and several dendrite enlarged here, cell body and the long axon. But conditional knockout, as you can see here, this is cell body stained with the neurofilament M axonal marker. This neuron extend several short abnormal axon. This could form recurrent circuit. Okay, this is a summary of the phenotype for KIF2A conditional knockout. So severe cortical and hippocampal focal epilepsy occurs. And that is because aberrant neuronal network formed in cortex and hippocampus. So, and also axon dendritic differentiation was affected in this knockout mice. So in summary, we've clearly identified or clarified mechanism of cortical malformation and also epilepsy in the KIF2A conditional knockout mice, and also in a patient which is affected with the KIF2A function. So I stop here. Thank you for audience.
Thank you very much, Dr. Taka. Very interesting talk. One question, as we did in this session, of Loboteka's choice. One question.
Okay.
That's very interesting. So, for the postnatal model, do you find the reason why it's only the cortex and hippocampus axons affected? What about the other axons?
Okay. Yes. The main reason is the KIF2A expression. KIF2A expressed hippocampus, mostly fiber, abundantly, and also, cortical neuron too. So in the cortex, it happens also. I didn't show the data. So that you can see the seizure in both hippocampus and the cortex. Okay? You understand? Okay.
Excellent. Thank you, Loboteka, for sticking to the time. Now, the time came for the new group leaders at the CGMR, Heba Al-Khattabi. She is a newly graduated PhD investigator from UK. She's going today to talk about RPL27A, a novel target for microRNA 595, and its contribution to meiosis via ribosomal dysgenesis. Heba, the floor is to you. Thank you very much.
Good afternoon, everyone. It is my pleasure to be here today to talk about the role of the ribosomal protein RPL27A as a novel target for microRNA 595 and its contribution to the myelopoiesis via ribosomal dysgenesis. Before I start, I would like to give a brief introduction about microRNA. MicroRNA are short, non-coding RNA which play an important role in regulating gene expression and several important biological processes. There are increasing evidence suggests that microRNA altered expression may contribute to different diseases, cancers, and hematological disorder, including a disease called myelodysplastic syndrome. Myelodysplastic syndrome, it is a disease of the elderly people, like in the average of 72 years. And it is characterized by anemia cytopenia, and it has a high risk of transformation to acute myeloid leukemia by 30%. Cytogenetic abnormality play an important role in the diagnosis of the disease, and they are seen in 50% of the cases. The most common chromosomal abnormality that are seen in MDS patients are abnormality in chromosome 5. And the second most common abnormality is seen in chromosome 7 as a monosomy 7 or deletion in the long arm of chromosome 7. And when the patient they are diagnosed with monosomy 7 or deletion in the 7q, they are characterized under a high-risk group or poor prognosis. To date, there are not much research that focus on the role of the microRNA localized on chromosome 7 and its contribution to the pathogenesis of MDS disease. Therefore, in this study, we focused on studying one of the microRNA localized in the long arm of chromosome 7 at the end of the chromosome, which is MER595. There were no any study that has been published before about this microRNA and its general function or its regulated or target genes. So to know what is the target genes of this microRNA, I have to apply like an assay, and due to the limitation of the available assays such as computational algorithm or the proteomic or microRNA, in King's College, Professor Job Jakin has developed a novel functional assay. Basically, this assay depends on using cDNA library representing 16 or almost 17,000 unique genes cloned downstream a vector which has a two selectable marker, confers GCV sensitivity and xeosin resistance. I will describe this methodology. So in the beginning of this assay, we transfect a cell line, which was easy transfectable, such as MCF7 cells, and this cell line doesn't express the microRNA of interest. We did around 25 transfection with the cDNA library, and then we start a zero-sen selection. So the zero-sen resistant cell then transfected with another vector which has the microRNA cloned in, and it has a puromycin sensitivity, and the puromycin resistant cells then selected with the GCB ganciclovir. The cells which has bind to the microRNA or has a target gene for this microRNA will be resistant to the ganciclovir, whereas the other cells which is not regulated by this microRNA will die from the GCB. I was able to isolate around different GCB resistant cells from around 21 flask out of 25 transfection. These isolated cells were PCR amplified and direct sequenced. The image here representing the different band size. Each band is expected to be a different target genes for this microRNA. The direct sequencing result and the blast analysis come out with the identification of RPL27A and HEAT-shock protein 14. There were several research that shows the important role of the ribosomal protein in the pathogenesis of the myelodysplastic syndrome. So I focused on studying the biological function of this ribosomal protein and its relation with the pathogenesis of MDS. But before starting anything, I have to validate if this is really a real target for MIR595 or not. Therefore, I transfect 4 different cell lines with the microRNA and with the empty vector as a control. And the result has shown that the microRNA down-regulate the target gene, which is the normal relation between the microRNA and the target gene. If the microRNA is up, the target gene should be down. And as you can see here, on the mRNA level, the level of the RPL is down, and the cells which is transfected with the microRNA compared to the empty vector. Also, on the protein level, I did the experiment only in 2 cell lines, and similar result was obtained if you compare the cells transfected with the microRNA to the empty vector cells. Therefore, we can confirm that this is a target for MIR595. So we would like to understand what is the function of this ribosomal protein. And it is already known that ribosomal protein played an important role in the MDM-P53 pathway. So in the normal condition, ribosome biogenesis takes place in the nucleolus, and the ribosomal protein helps or aids in the assembly of the ribosome. So, under this normal condition, P53, which interact with the MDM, undergoes ubiquitination and degradation, and then it gives us a normal cell proliferation. But if there is a ribosomal stress result from disruption of ribosome synthesis, we will have free ribosomal protein that could bind to the MDM and prevent the binding to the P53. And that results in cell cycle arrest, DNA repair, apoptosis, and senescence. And there are several microRNA has shown their, ribosomal protein have shown their role in this pathway, and we would like to know if RPL27A is acting similarly or not. So, to identify the function, we did a knockdown using a lentivirus on several cell lines that express the P53 and cell line that doesn't express the P53, and infecting those cells with the lentiviral shRNA for RPL27A. And I was successful in getting a high knockdown level, around 80% by one of the shRNA and around 50% knockdown by another shRNA. Therefore, I moved further to apply my biological function. So to study the relation with the P53, I did the screening for the P53 genes. And I did that on 4 different cell lines, the HCT116 wild type and the double knockout model, and the health cell line, which is erythroleukemia cell lines, and K562 myeloid cell lines, which is null for P53. And the result has shown an activation of P53 and the P53 expressing cell lines. In addition to that, the P53 target genes such as P21 and PAX was also significantly upregulated, either by looking at the mRNA level, and this is the protein level showing the activation of P53 in both P53 expressing cell line, and looking at the null P53 cell lines such as HCT double knockout K562, no P53 definitely, but we can see no any changes on the P21 or PAX. So to understand or to know if the effect of the P53 activation has an important role in the apoptosis, or does it induce apoptosis or not, I did the annexin V5 staining, and it is also significantly shown that the knockdown lead to apoptosis and the P53 expressing cell line as well as in the null P53 cell line, and that was surprising. And even though the level of the apoptosis is less than what was seen in the P53 expressing cell line, but it was significantly increased level of apoptosis. And looking on the other cell lines, the health cell lines which is expressing P53 and K562, you can see there is much increases in the apoptotic cells compared to the wild type and the empty vector cells. And this is another negative myeloid cells for P53, and it is very clear here the increases in the apoptotic level. So to understand why we are seeing this induction of apoptosis in the null P53 cell line, we propose that there is a disruption in the ribosome biogenesis. So looking at the ribosome profile, doing sucrose fractionation on 10 to 50% sucrose, we can see that RPL27A, which belonged to the large ribosomal subunit, has led to the reduction in the 60S subunit compared to the control, the wild type cells or the empty vector cells. And if we look at another positive control from the small subunit, we have a reduction here in the small subunit. And when we look at the null P53 cell lines, also we get the same result. And that conclude that the induction of apoptosis which we have seen in the null P53 cell lines is due to the disruption of the ribosome. Also looking at the nucleolus and examining the effect on the nucleolar, we did the confocal screening using fibrillary, which is a marker for the nuclear, and it is very clear here that the knockdown with RPL27A or the other positive control led to the disruption of the nuclear compared to the empty vector or to the uninfected cells. Okay. If we go to the cells again, and if we did the same experiment, but if we did our analysis on 48 hours instead of 6 days period, we can see that all the effect is a P53 dependent, which like either by increasing the apoptosis or the significant reduction in the cell proliferation and the reduction in the 60S subunit, which was not much as what we have seen in the day 6. And that means what we have seen on day 6 is effect of ribosome disruption, and which we can see it as early during a 48 hours period. To confirm our result and to see if it has any effect on the hematopoietic stem cell, I did similar experiment or knockdown experiment on CD34 cells isolated from peripheral blood mononuclear cells using several different cytokines, and I did a knockdown at 24 hours after the isolation of the CD34, and after 6 days I did the RNA and protein extraction and colony forming assay, and the rest of the cell undergoes a differentiation. I tried to induce erythroid differentiation to see if this knockdown has any functional effect in the erythropoiesis. Similar to what we have seen in the cell lines, we see it again in the CD34 cells or hematopoietic stem cell, activation of P53 and the reduction in the cell count. Looking at the effect after 11 days from the infection and looking at the effect on the erythroid differentiation, by 11 days And the live dead staining showed that most of the cells which are transfected with the lentivirus against RPL27A are dead. And looking for the only survived cells and stain them with the two marker for erythroid, the CD71 immature erythroid and the glycophorin, the mature erythroid. We can see that in the mock, the uninfected cells or the cells infected with the control, the differentiation is going well and majority of the cells goes to the maturation. But with the RPL27A or the other positive control, the majority of the cells are not differentiated properly or doesn't undergo erythroid differentiation, which means that this knockdown is affecting the erythropoiesis process. Finally, we did a small screening on a small number of patients to see if this ribosomal protein or if the microRNA has an important role or any contribution in the pathogenesis of the disease. I screened 27 MDS patients, and we classify them in a two way. The first classification was based on their cytogenetic abnormality, and there were around 11 patients with monosomy 7, 5 patients with del5q, 6 patients with a complex karyotype including mono 7, and there were patients with a normal karyotype. And we can see here that in the monosomy 7, the expression of MER595 was significantly downregulated compared to the patient with a normal karyotype. Also, the patient with a complex karyotype, including abnormality in chromosome 7, was significantly downregulated compared to the normal karyotype. And for the ribosomal protein, the vice relation also observed, but this time it is with the 5q minus, which is considered as a good prognosis, similar to the normal karyotype. The other classification was based on the IPSS scoring, which classified the patient either as a high-risk group or low-risk group. And we have around 18 patients classified as high-risk, and monosomy 7 or abnormality in chromosome 7 comes under this group. And the low-risk group are 9. And you can see here there is a significant difference in the expression of the microRNA between the both groups. It is down in the high-risk group, and the RPL is up in the high-risk group. So to summarize, this is the first study to show that microRNA 595, which is localized in 7q36.3, is an important regulator of ribosomal protein 27A. In vitro work has shown that both p53 dependent and independent effects following RPL27A downregulation or deficiency was seen, which also include inhibition of cellular proliferation and induction of apoptosis and defective ribosome biogenesis. Also, this is the first study to show that the knockdown of RPL27A impaired the ribosome biogenesis. From this study, it is evident that RPL27A depletion led to a severe cell death, and it has an effect on the maturation of erythroid or in the differentiation of erythroid cells. Lastly, we showed that for the first time that MER595 appears significantly downregulated in the majority of the primary samples derived from patients with monosomy 7, and also when MDS high-risk cases are compared to the low-risk cases. These findings have a valuable impact in understanding the disease pathogenesis in monosomy 7. I would like to thank my supervisor from King's College London, Professor Ula Mufti, and Dr. Yob Jakin, who has developed the functional assay, and also the Department of Hematological Medicine. Also, my thanks go to the Center of Excellence of Genomic Medicine Research for funding this research, and to King Abdulaziz University. And also, I would like to thank Professor Alan Warren from Cambridge University, who allowed me to perform the ribosome profiling experiment in their lab. Thank you.
That's great. Thank you, Hiba, very much. We will allow one question. One question. If there is no, okay. Have the microphone for the list.
Well, thank you
this nice work. My question, the simple question, is this mere RNA that you're talking about, is it biallelic expression or monoallelic?
Sorry, I can't understand your question. What do you mean by
My question is, since you are dealing with monosomy 7,
yes.
Is it mono expression or biallelic expression?
No, mono. Yeah, mono.
So it's mono, that means you are already getting,
Yeah.
low level of the.
Exactly. Yeah.
Thank you.
As a privilege for the ladies section, we can have one more question from ladies section, if there is any. If there is none, then we'll move to last two sessions, two talks. But Prof. Ali would like to do an announcement, quick announcement.
There's an important announcement regarding the organization. For tomorrow, from 1 o'clock till 2:20, there's a poster session. So all the poster presenters are requested to kindly stand next to their posters, because the evaluators are going to pass by and the best posters will be selected. So all the poster presenters, please, between 1 and 2:20 tomorrow, stand next to their posters. After 2:20, the exhibition will be open to ladies only. After 2:20. Thank you very much.
For the technical assistance, please, can you switch from the ladies side to our, yeah. For the next speaker, it's affiliated, distinguished Professor Emmanuel Belmezakis. He is a professor of genetics, Department of Genetic Medicine and Development, University of Geneva Medical School, Switzerland. Emmanuel is interested in the genomics of gene expression and cellular phenotypes in human population and multiple tissues, and how this assists in the interpretation of regulatory networks and human disease variants. Today he's going to talk about population and personalized genomics to understand the human biology and diseases of the.
Okay, well, thank you very much, and it's a great pleasure to be here again in this conference and in this university, and with colleagues that we interact frequently, either in person or from distance over Skype and telephone. What I'm going to try to talk to you about today is some advances that we've made through a particular project, but I'm going to sort of extend it a little bit on work that we've been doing in our lab over the many years, on how we can bring knowledge of genetics at the molecular level in order to be able to understand and interpret signals at the disease level. I think that's the, yeah, okay. So, thinking about revolution in medicine, I think many of us are, some more than others, are thinking about how is the next revolution in medicine is going to go? Where is it going to come from? And clearly, revolution in medicine is coming from advances in technology, all kinds of medical devices that allow for better interventions, better diagnosis, and so on. But one of the things that we have been missing over the many, many years is deep understanding of the human body, of how the human body works. And technology can go so far into being able to intervene if we don't really know how the human body works and how that intervention is going to have consequences on specific physiology and other aspects of the human biology. So what I'm arguing, and I think that's not a very difficult thing to convince, hopefully, is that the next revolution in medicine is going to come from deep understanding of human biology. There's many things that MDs do now, I'm not an MD, that are a little bit sort of blind. Many drugs, we don't even know specific mechanisms. They just happen to work and then we keep using them. And we only hope that there's no sort of long-term side effects. But better understanding of human biology is not only going to allow us to understand better what we've been doing so far, but also understand where we should be intervening with what kind of interventions, what kind of medications or even lifestyle changes and so on. Now, one of the big problems that we have from the genetics perspective, and that's me as a geneticist, is that we generally look at this genome that we inherit at -9 months of our life. And what we're trying to do is we're trying to build relationships with organismal phenotypes, diseases that usually manifest themselves quite late in life, maybe when somebody's 40, 50, 60, even 70. And one of the ways that we've been thinking about it, and I think the most conventional way to think about it, is that there's DNA information that does something. And the reason that it's late onset is because it's the environment that is accumulating its effect over time that together with the genome gives rise to a particular phenotype. Now, I want to add another component of it that has been given less attention, which is the exposure to our genome. So we have been thinking of exposures to the environment as a long-term time-dependent effect. I think there's a good reason to be thinking of genetic variants as a long-term exposure that cumulatively leads to a particular phenotype. So, for example, if you think of it in sort of a naive way, if you have a hepatocyte that has, actually the genome has, but there's a mutation in the genome that affects specific functions of hepatocytes, you can imagine that it's a small effect if you look at it over an hour, but if that same thing happens, this suboptimal function happens over 30 years, that's going to have substantial consequences on certain cells. And it's even worse when you're thinking about cells that are not dividing, because there the cell doesn't have the ability to sort of reprogram itself or change whatever memory it has from this exposure of the suboptimal effects of functions that we're thinking about. So in a way, we have to be thinking the time-dependent effect, we have to be thinking more, not only in terms of the environment, but also in terms of the genome, and jointly, either by interacting or independently, how can they give rise to particular phenotypes. Now, the genetic information as well as the omics information that comes from, let's say, transcriptome, proteome, metabolome, and so on, has been given a lot of attention because people are interested in understanding that deep biology that I was talking about. Now, however, the genomic information, the genome itself, has not been able to provide the information to predict somebody's risk at birth. So it's very difficult to say, for example, whether somebody will develop diabetes on day 1 of their life. Now, we can somehow try to use this information in different levels without necessarily saying that the genome is not useful for prediction. And here's how to think about it. So clearly, understanding aspects of disease causality, including genetics but not exclusively, will give us clues about what kind of drugs we might be thinking of using, what kind of interventions we might be doing, as well as of environmental effects that might be relevant in terms of lifestyle changes that have an impact on the phenotype. That's something that more and more is sort of the intention and the objective for genome-wide association studies, when we're really not going to get the predictive value that might have been anticipated early on. But the second thing is that we can start really getting into individualized risk assessment, not from the point of view of birth risk assessment, but from the point of view of understanding somebody's risk much earlier than we can do it right now. So if instead of, for example, of assessing somebody's cardiovascular risk when we look at lipid profiles when somebody's 40 or 50, and when they get abnormal, then we can say that somebody's at risk, we can probably look at omics profiles like other types of metabolomics or transcriptome, combined, of course, with the genome, that gives us clues about this much earlier in life, which means that there's more opportunity for intervention. Sometimes lifestyle, sometimes pharmacological. Now, in order to put all these things together, we need to start, we can do it in 2 different ways. One way is to do this machine learning approach, where we just throw everything in some kind of computer, so this kind of black box way, and then learn something, which is going to be highly overtrained, highly specific to the data set that we have, and is not going to give us mechanisms and understanding of biology. The alternative way is to try to piece all these things together in a biological framework where we start to understand how these particular molecules interact, in which context, in which tissue, at which time point. The phenotype, I mean, the 3 alternatives would be that DNA is impacting the phenotype, which is what I'm arguing is the right direction. The other one would be that the phenotype is impacting the genotype. That doesn't happen. You cannot change your genotype because you get diabetes. Third one is that it's parallel. But it cannot be parallel because the genome is established much, much earlier than any phenotype of an organism, especially the multicellular complex organism of humans, is established. Therefore, it's kind of like an axiom that we accept that genome to phenotype association is a causal relationship. Now, the specifics, we have to figure out which nucleotide and so on. So in order to start, we have developed strategies where we look for what we call expression qualitative trait loci, where we look at genes as phenotypes. So we measure gene expression from, in this case, RNA sequencing, and then we associate nearby SNPs, exploring the cis-regulatory landscape, or faraway SNPs looking at the trans-regulatory space. And the trans-regulatory space, you can imagine it as a SNP that impacts a transcription factor, and then that transcription factor, the levels being, let's say, modulated, impact the targets of that. So you can have an association like that I showed you before, where there's a correlation between the level of expression, in this case, and the genotype, and it's basically a regression, it's a correlation between the number of copies of C. But actually, the direction of that is kind of irrelevant. It's completely conventional. Now, why do we put so much effort and so much focus on looking at these expression QTLs? Well, we do that because about 90% of genome-wide association studies has been shown to reside in known coding sequences. And as Steve said before, 99% of our genome is known coding. So there's a lot of information there that is going to give us explanation for disease that we haven't been able to explore simply because we don't understand the known coding component of the genome. So here's an example. I have this region here. Well, I have a SNP that is associated with a disease, let's say diabetes. If you try to understand just very simple things, this is a SNP. What does it do? Just basic molecular information. What does this SNP do? The first thing to think about is that it does something to genes nearby. And the question is what and which gene? Okay? Because the SNP is here, there's no information as to what it does. And you cannot assume that it's doing something to the next gene 3', like for example this one here, because regulation doesn't work this way, it's quite complex. This SNP could be affecting actually a gene right down here, 2 or 3 genes away from the SNP position. So unless I know the mediator of that effect, the gene, I have no idea, I don't have a handle on the biology that is associated with this association that I found. So what we need to do is we need to understand what does a SNP do, and because it's most likely going to be regulatory, I want to explore how does it affect the expression of nearby genes. And there have been methodologies that try to integrate expression QTL studies with genome-wide association studies to ask the question, am I looking at the same causal variant or different causal variants so that I can use the eQTL as a hypothesis? But there's additional problems because genome-wide association studies are done on DNA, and you just correlate an organismal phenotype. eQTLs are not done on DNA, they're done on RNA, and then the question is, which RNA, from which tissue? There's more than 200 cell types in the human body. Of course, it gets more complicated, probably more than that. And so depending on which tissue you look at, or which cell type you look at, there's going to be a different expression pattern. There's going to be a different effect of that variant on regulation. For some variants, it will affect one gene. For some variants, it will affect another, or none at all. So the tissue that you're going to be looking at for RNA is extremely important. And also, what is important is from the perspective of the disease. If you're interested in diabetes, what tissue will you be looking at? Islets, pancreatic islets? Will you be looking at liver? Would you be looking at brain? What about other types of tissues? What about muscle or artery or heart muscle or whatever? Which one is relevant? And by relevant, I don't mean affected, I mean causal. So something that happens in that tissue predisposes somebody to have a cardiovascular event. This question is or a diabetic event, or getting diabetes. It's very, very difficult to assess what's the causal tissue because it's not something that is readily obtainable from the data. We cannot get human tissues just easily. I cannot just dissect every one of you and you remain living and get 30 or 40 tissues. And we need large sample sizes, which means that we need a systematic effort to be doing that. Just to give you one of these caveats, you can have a case where, again, bring in the same region that I showed you before, two tissues, pancreas and liver, let's say pancreatic islets in this case, they are the same EQTL, the same SNP, affecting the same gene in both tissues. But which tissue is mediating the effect on disease? Is it both? Is it one of them? It is important to know, because if I want to understand network effects, I need to be looking downstream effects in the correct tissue, even if the gene is the same in both tissues. Or I can have this effect where the same SNP is impacting two different genes in two different tissues. Both appear relevant, liver and pancreas, but I'm not going to know which one unless I'm able to add additional information. So we need an unbiased way of looking at the human body, rather than a biased way that looks at only what we call relevant tissues. And to do that, there's this project called GTEx, which was started a few years ago, which explicitly samples from post-mortem donors about 30 tissues, and the idea would be to get to about 900. We're midway now to 450. This will be done with RNA sequencing. There's whole exome sequencing, whole genome sequencing for the same individuals, and there's going to be additional assays, what we call enhancing GTEx that go into methylation, proteomics, DNase I representative sites, and all kinds of other things. So what we want to bring is all these tissues now in what I said, kind of a more unbiased setting. Again, it's not 200 cell types, but it's 30 or 40, actually close to 50 now, where I can discover EQTLs and I can start building these associations with genome-wide association studies and have a more unbiased interpretation scheme. This is the figure from the pilot paper, which was published last May, where you see the number of EQTLs by sample size for the 9 tissues for which we had a good sample size to discover EQTLs, which means above, I think this was 60 or 70. And as you can see, the numbers are, depending on the sample size, anywhere from 1,500 to 2,500 EQTLs per tissue. We started building some more kind of manually curated links between genome-wide association signals, like this one in blood pressure with 2 genes, which have not an appreciable level of expression in artery, in tibial artery as we get here, but actually a strong effect with respect to the EQTL relevance to interpret the GWAS study. But more specifically, we developed a few years ago a method where we statistically infer that correlation, and we can test that correlation between the GWAS and the EQTL to see if they overlap, if they're mediating the same effect. I'm not going to go into technical details, it's already published. And what we can do now is we can apply this in the 44 tissues of the GTEx project that we have available in this midway data, the 450 individuals. If you do that, you get a strong correlation of how many of the GWAS signals you can interpret, you can correlate with EQTLs, which is highly correlated with sample size. Even when you correlate we correct for the number of EQTLs that you have per tissue, because when you have more EQTLs, you're more likely to capture a signal. The reason that it correlates with sample size is because you have more rare recombinants, which disentangles the correlation of the genome. And when you do that, you can start getting this kind of patterns where you basically get a number of tissues here and a number of conditions here, and what you plot is the enrichment of the relevance of one tissue to one disease. So basically, where do I get an excess of interpretation, an excess of overlap of GWAS signals for, let's say, narcolepsy with some of these tissues? And you can start building hypotheses, not about individual signals now, but about tissue causality, the participation of individual tissues. I'm not just going to skip this a little bit faster, so here's some of these profiles which are currently unnormalized for sort of the random expectation for 44 tissues for body mass index, type 2 diabetes, and schizophrenia. But just to give you an idea for what comes up, let's say in type 2 diabetes, you get adipose tissue to be quite enriched, tibial artery, and several brain tissues, which is not surprising, especially for almost all of them. Tibial artery is a little bit off, but we have to see what that means. And you can go one step further by saying that I can get the average contribution of tissues if I look at all variants discovered in the population, but not all of us carry the same risk variants for diabetes. So then I can ask the question, for the 30–40% of us that carry a risk, 30–40% of risk variants that each of us carry for a particular disease, where these are active. So you may find, for example, that for some individuals is this tissue, which is, I think in this case, is liver, or for other individuals it could be pancreas. Which means that although you can get a contribution to the population, you can also have personalized contribution of these genetic effects in tissues. Finally, you can use GTEx as a reference where you have all these tissues, you have a few expression values for your cohort of interest with a lot of clinical information, and you can use imputation methods to infer expression patterns in tissues for individuals which you have for tissues you have never sampled. So you can get, for example, blood, skin, fat, some other easily accessible tissue like muscle, and get an expression profile which is not going to be perfectly accurate, but it's going to be close enough for brain, which you can never sample from a living individual. So to summarize, I think we have an unprecedented catalog of regulatory functional variants in the human genome. We get a first glimpse of these effects in diverse human tissues. We can turn this tissue activity into a tissue causality profile, and this tissue causality profile can become, implicate pathways processes, but also can become individualized at the level now of the tissue, not at the level of individual signals. So with that, I want to thank the GTEx Consortium and of course my laboratory and the funding agencies. Thank you.
Where are you?
Behind one of my students on top. Just a little. I am young. What do you mean?
All right. One question, please. One question.
There's one over there.
Yeah, I have actually just a few small questions.
Excuse me.
It's really, I think it's very complicated data. Do you have a sort of software or program designed just for as a bioinformatics for your project? This is one. Second, how far into you are into the project, and how long do you think you need to come up with informative results that can be helpful?
I think the project is basically a long process. It's not going to end, in a year. We're going to get some clues in the next sort of few months. The GTEx project itself is going to finish at the end of 2016. We are going to launch. We're writing currently a white paper to launch an international GTEx project. It's one of the ways that we can. One of the values of the GTEx project is not just about the genetics of gene expression, but also the diversity of gene expression in multiple tissues given the diversity of environments. If you sample 900 individuals from the US, the diversity of environment is not going to be that much. But I'm pretty sure that if you sample individuals from the US, Saudi Arabia, China, Sub-Saharan Africa, Europe, you will get such a diversity of environmental effects combined with the diversity of the genome that you will get some pretty advanced picture of the human body biology and how it's affected by that diversity. So the idea would be to do this for 20,000 to 30,000 people worldwide. Now, for the bioinformatics tools, it depends on which level you want to automate that, or you want to do a research project, which means that you have to take pieces that you put together yourself. But there are some of these that we have produced packages for that we can share.
Thank you. Right. As a courtesy for Steve, one more question from Steve.
So,
yes.
Just trying to understand. Should I go ahead?
Yeah. Yes, Doctor.
Go ahead, yes.
Yeah, I'm a genome student. I'm still not graduated yet. And I have a question here.
Yes.
I was wondering about you said the human tissue types of QTLs, it's hard to obtain. I was wondering if you can use some tissue culture or maybe use some stem cells too, so we can obtain some of these tissues?
That's a very important question, very interesting question. Yes, you can, and no, you cannot. It depends on which way you want to see that. If you are thinking of sort of a purely genetic effect, and if you had a perfect way to transform any sort of, let's say, stem cell or iPS into the appropriate cell type through differentiation process, you would get a better answer than you get here just for genetics. The disadvantage is that you get no environment because by definition, the IPS should have removed all the environmental exposures of that individual, by definition. It won't, but then it would be random what has been left. So, it would be great to have a bank where we generate cell types from IPS, but that's not going to give us the full range of biological effects that we're looking for.
Okay, Steve?
So you're using GWAS data.
Mm-hmm.
Have you modeled how deep you can go on the type and number of variants that are influencing EQ2?
You mean number in terms of
Well, I'm assuming you're
frequency?
using GWAS SNP data.
Yeah.
I'm assuming you're also doing CND, but could you go deep into multifactorial disease? Can you model 3 or 4 loci, for example, effective 3 or 4 loci?
Well, we can. I mean, that's the whole point of doing this at the tissue level rather than at the level of a single variant, because...
Saying, can you do it? You have an example?
We don't have an example because we haven't advanced that much, but that's the point. The point is to do this in a system-like fashion rather than in an individual variant, which is, and I believe that the first level of that system will be the tissue. The second level will be the organ.
Okay, thank you very much for that. Thank you. It was very interesting. Thank you.
Excuse me, one more question, please.
All right, okay.
What's the criteria used to determine the causal tissue? Like in diabetes, it's a syndrome. It's multi-organ disease. Okay, so
yes.
How do you decide that which is the causal tissue?
Well, there's not going to be one causal tissue. There's going to be multiple. The idea is that if I have, for example, let's say 100 variants for diabetes, which are known to be associated, I'm going to try to find in which tissue the activity is present that is also causal for the increased risk. And if I see, for example, a particular enrichment in one tissue, like you would expect for liver, for pancreatic islets, for brain, for example, then you would put a prior of that tissue to have a significant effect. And of course, that's going to be, in general, an average in the population that has a certain contribution, and it's going to be much more skewed at every individual because every individual has a different set of risk variants. I'm not sure if I covered you, but it's not an easy question to answer in the 10 seconds. And
thank you very much. I think we're going to stop questions here. Thank you very much, Emmanuel. Now the floor is to the ladies section again, and this time with Professor Anne Goodeve. And the first time she visits and speaks in the IGMC. So welcome to her. Anne is from Sheffield Diagnostic Genetic Service, Sheffield Children's NHS Foundation Trust, from Sheffield, from United Kingdom. And Anne is going to talk about her experience with the NGS as analysis advanced tool in her lab. The title is Experience of Next Generation DNA Sequencing Analysis in a UK Genetics Laboratory. Down to you, Anne.
Thank you very much. I'll just try and get the slides up. Okay, I'll start by thanking the organisers very much for the opportunity to come and visit Saudi Arabia. It's my first visit and I'm very happy that I've been able to catch up with some of our previous PhD students from Sheffield while I'm here. So I'm going to talk on an experience from part of our genetics diagnostic service. So I spend half of my time working at Sheffield Diagnostic Genetics Service as a clinical scientist. I'm a member of staff of about 100 within our service, and we undertake both molecular and cytogenetic analysis within the service. And I look after an area that includes bleeding and thrombotic disorders and some of the inherited anemias particularly. So I'm going to talk about a couple of our next generation sequencing panels to you today. So the aims of this piece of work were to introduce next-generation sequencing panels for Fanconi anemia and for coagulation and platelets gene disorders. And we started next-generation sequencing in our service about 2 years ago. So the first disorders that we introduced were for connective tissue disorders and for inborn errors of metabolism. And over the last couple of years, they've been followed by about 10 other panels for specific types of disorders, with between about 10 and 100 genes in those panels. So, a small bit of introduction just to these two sets of disorders. So the coagulation and platelet bleeding disorders can often be diagnosed through phenotypic testing undertaken by a coagulation laboratory, which means that we often have sufficient information to be able to target one or a small number of genes associated with a particular disorder. Fanconi anemia is the second disorder that I'm going to speak about, and that can result in bone marrow failure. And there is a phenotypic test that can be undertaken, which looks at chromosome fragility testing, and this may be undertaken prior to genetic testing to confirm an individual's phenotype. But in our experience, it's done fairly rarely, so the phenotype often hasn't been pinned down that well when the patients are referred to us. So I'll start with the hemostatic and platelet gene list. So these are mostly bleeding disorders, but not entirely. So ADAM-TS13 causes a thrombotic disorder, whereas most of these are associated with bleeding, apart from the three fibrinogen genes, which can be associated with bleeding or with thrombosis. And in addition to being able to analyse any of these genes independently, we can also group them where we have maybe an unknown disorder. So for example, for factor 8 and factor 9, if we have a family with a history of haemophilia, but we don't know the type of haemophilia because the male who's affected is long dead, then we can analyse both factor 8 and factor 9 simultaneously. If we have an individual with a reduced factor 8 level, but insufficient family history to be able to say whether it's likely to be X-linked haemophilia or autosomally inherited type 2N von Willebrand disease, we can analyse both those genes together. We can analyse both genes for factor 13 deficiency simultaneously, all of the genes associated with the fibrinogen disorders, and both of those associated with Glanzmann thrombasthenia together. And this has enabled us to speed up our diagnostic process. These are the machines that we use. So we initially started out using the Illumina MySeq, and over the last year have introduced use of the HiSeq. So generally we run the HiSeq machine about once a fortnight, and we try to fill that up with 96 samples where possible to make best use of the machine. The data analysis then takes a couple of days to reach us. So this is an example of a very simplified example of the workflow that we use. So we follow the gene analysis toolkit designed by the Broad Institute and use a slight modification from our in-house bioinformaticians. So we start with all the variants that were identified on the panel, limit them then to the variants within our genes of interest, and generally with use of this panel we may only be looking up to 3 out of the 13 genes. Any of the frequent variants noted on the sequence variant databases without any pathogenicity attached are removed by the analysis pipeline. So all the top part of this analysis is undertaken through the bioinformatics pipeline, and then the variants identified at the end of that process that may be pathogenic are analysed by our clinical scientists, including myself. We then undertake a pathogenicity assessment that I'll come to in a moment, and end up possibly with a small number of variants. So for haemophilia, probably only one for some of the autosomal disorders, possibly 2 variants at the end of that process. So it's one of the simplest pipelines that we can have for our next generation sequencing. So the top example on here shows a patient with haemophilia. So we've got the defect in the factor VIII gene. It gives us the reference sequence, the exon, the nucleotide, and the amino acid substitution. Shows the genomic location for that variant and the original and the variant nucleotide. And these further examples just show an individual that we analysed for a fibrinogen disorder, and the variants identified, with the lower one being a common pathogenic variant. So once we've undertaken our next-gen sequencing, we use Sanger sequencing to confirm any potentially pathogenic variants. During the validation of the hemostasis panel, we reanalysed a number of patients that had previously had Sanger sequencing undertaken. We only analysed the genes of interest which had previously been investigated. And our criteria for having full coverage were that we had 30-fold sequence coverage for our regions of interest, which were the exons plus or minus 5 base pairs, and then 18-fold coverage at least for the flanking intronic sequence out to 25 base pairs. And we had complete concordance of the variants previously identified in those individuals, which were between 2 and 30 variants per individual, with the 30 variants being in von Willebrand factor. We were particularly interested in von Willebrand factor as it has a partial pseudogene, so the coding genes on chromosome 12 and the pseudogene on chromosome 22. And it covers a region of about 13 exons of von Willebrand factor in some of the main binding sites for von Willebrand factor. So we made sure that we included 5 patients within the analysis for this region, and these 7 variants shown were identified in the 5 individuals within the pseudogene, within the region that's copied in the pseudogene. And these 2 regions have got a 97% sequence similarity. But we were able to identify the 7 variants previously seen on Sanger sequencing and didn't identify any additional variants within that region in these patients. And subsequently, we've been able to identify 2 gene conversions in this region, which are where part of the pseudogene sequence invades the coding gene and introduces changes into it. So we're very happy that we can uniquely identify the von Willebrand factor coding gene without interference from its pseudogene. For our pathogenicity analysis, we follow the recommendations of the UK Clinical Genetics Science Association. We document all our variants using a so-called unclassified variant form. We start with our locus-specific databases to see whether any variants have been identified in our gene of interest, and also use the human gene mutation database. We look at variants identified on the exact exome sequencing project and the single nucleotide polymorphism database as well. If necessary, we undertake in silico predictions using the software Alamut, which provides links to a number of different software tools. And we also have an in-house tool referred to as SNPbot, which has been designed by one of our bioinformatics trainees, and it links to several of the online tools to help with pathogenistic predictions, particularly for amino acid substitutions. And the link at the bottom is to the recommendations that we use. So the patients analysed to date for the hemostasis panel are shown on this slide, and the ones which we look at particularly commonly are those for the haemophilias and for von Willebrand factor. So they're our main genes that we analyse on a more regular basis. And you'll see for a number of the areas that the pickup rates are good for mutations in these disorders because of the phenotypic analysis that's already been undertaken. Our overall detection rate for the mutations in these individuals is 70%. So our impact on the bleeding disorder testing is that von Willebrand factor, which has 52 exons, and we previously undertook by Sanger sequencing as 55 separate amplicons, is now much more straightforward. And rather than analysing specific regions of the gene associated with particular phenotypes, we just look at the entire gene in virtually all of the patients referred. We can look at carriers of unknown haemophilia types where we haven't got an affected male available in the family. And we can look at disorders such as 2N von Willebrand disease and mild haemophilia A together, rather than having to look at the genes sequentially. And where there's more than one gene associated with the disorder, we can just analyse the lot together. I didn't mention, but on the previous slide, one of the individuals that we were able to look at was a baby that had died shortly after birth of the bleeding disorder. And we were asked to look at a number of different possible causes for that, and we're able to just look at all the 5 genes that they requested at the same time, speeding up the analysis. So very briefly on Fanconi anemia. This is an inherited disorder, generally autosomal recessively inherited in all but one gene. It has features of short stature, cafe au lait spots, often thumbs that are poorly attached and non-functional, and other limb deformities in several of the affected individuals. And as well as its features that I've just mentioned, often abnormal gonads as well, and microcephaly, so a small head. So in Fanconi anemia, it can lead to bone marrow failure and cancer susceptibility, and patients are often referred to us for genetic analysis to look at the cause of the bone marrow failure in that individual, and is it due to Fanconi anemia. So we can attempt to identify the causative mutation in the index case, look at relatives subsequently where we can identify a mutation to see whether they're relevant as a bone marrow donor, and also look at relatives to see whether they have an elevated cancer risk because of some of the genes that are associated with the disorder. This is the current list of the 16 genes that we analyse at the moment. And a number of these you may recognise as being associated with a cancer predisposition, such as the BRCA2 gene. This next slide summarises our findings to date using the next generation sequencing. So the majority of the mutations that we've identified are in the FANK-A gene, which has a predominance of mutations for the disorder. But we've also identified mutations in a further 7 of the genes. The majority are homozygous or compound heterozygous individuals,